invited here, and I want to sort of talk twofold about my area of research, and that's molecular genetics. First, I just want to talk about the general advances in this field. I'm sure some of you are working in molecular genetics laboratories and are going to be familiar with many of these concepts. Others might not be, so I want to take you to just a few minutes sort of introduction to what basically has now become the age of genomics. Human genome sequencing and other organism sequencing is advancing so extraordinarily rapidly, it's really hard to keep up with the pace. Less than 10 years ago, the first human genome was sequenced. Actually, two different human genome projects competed with each other, speeding each other along, completing a human genome. That was announced in the year 2000, though that was sort of a rough draft, and it's been continuously improved. We're actually, it's like a computer operating system. Right now, I think it's version 36.1. Every few months, as they find errors, small errors in the 3 billion base sequence, they update it, make corrections, and so on. So it's a continuous process. Not only have we sequenced the human genome, a few years later, in 2005, the entire chimpanzee genome was sequenced. And this was very, very important, because you may have been comparing fruit flies or mice to human genes to try to understand their function. But by having our closest living relative, organism that we shared common ancestry with 6 million years ago, we can really get at what makes us human. Though initially, when we only had those two comparisons, we didn't know necessarily what made us human versus what made them chimpanzees. Is all we could see was the difference. So it required more sequences, and more recently, when the macaque genome was sequenced, an old world monkey, we were able to put the human and the chimp question into context. By comparing it to an even more distant relative, we could see specifically which genes changed and perhaps made us human. We're going to be able to eventually elucidate the genes responsible for language, cognition, and all these things by doing these close comparisons of our closest living relatives. And in an anthropology department, that's the kind of research that we take care of. So we've done a lot of looking at all of the whole genomes themselves, as well as individual genes, again, to try to figure out who's related to whom, when did they last share common ancestry, and, as importantly, what changes occurred since that common ancestry. And so a lot of the things I'll be showing in a few minutes are evolutionary trees, showing how we can try to figure out where changes occurred in the evolutionary history. If we can tie those changes to changes in the protein molecules or in enzymes structure or an RNA molecule, we may be able to, again, figure out what changes are so important. These very same changes are used in the disease process. So if we understand how these proteins work and have evolved, we'll better understand disease and hopefully be able to ameliorate those conditions. In the last several years, many more genomes are either finished or about to be finished. So the gorilla genome project is nearly done. The draft of the orangutan and gibbon have already been completed. New World monkey genomes have been done. Malagasy lemur and mouse lemur genomes have been completed. All told, there's going to be about 25 different primate genomes within the next 2 to 3 years that have been completely sequenced. We will be able to look at any gene at any stage, if you will, in evolutionary history and try to reconstruct their states. And again, once we can do that, we can get at their function. Genomics has moved along so fast that just recently, James Watson, the actual co-discoverer of DNA's entire genome was sequenced, And just within the last year, Archbishop Desmond Tutu, as well as Hassan Bushman from South Africa, have their entire genome sequenced. There's now a Chinese genome and a Korean genome, and other multiple human genome projects are being carried out. My own research, I'm actually, right after questions, I have to run back to NYU to put in a grant proposal. We're actually proposing to sequence 10,000 human genomes over the next 6 years in a major giant genomic sequencing project. Some of you may have heard that the Neanderthal genome is the most recent genome sequence. 40,000-year-old, well-preserved Neanderthal bones from Croatia actually yielded enough DNA to sequence the entire genome of the Neanderthal. We now know, for instance, that some Neanderthals actually had red hair and lightly colored skin. But it's not the same red hair and lightly colored skin you might think of somebody from Northwestern Europe. In fact, it's a completely different set of mutations that yielded the same phenotype. And so we're going to be able to reconstruct many of these extinct organisms and our collateral relatives in much greater detail, because we're going to be able to actually look at their individual proteins, express them in bacteria or other vectors, and carry out all sorts of interesting research using this genomic information. Here's another source of genomic information. Not only can we get DNA out of 40,000-year-old bones, my lab, in fact, specializes in getting DNA out of poop. It turns out there are enough cells left behind of the organism that shed these, and this happens to be a juvenile, female, a blackback, and a silverback male gorilla, collected by one of my grad students in the Congo, that we can actually get their genetic information out of them. For my field of research, working on very endangered primates, many near extinction, some of which are also extremely dangerous, you don't want to walk up to a gorilla and stick a needle in its arm to try to draw blood, believe me. But you can follow behind them, and after they get up from their nest in the morning and walk off, you can find these left behind, and you can sequence all of the DNA you want non-invasively. And I'll come back to this point in a minute, in carrying out conservation genetic research without having to bother or harass the animals at all, potentially changing their behavior or whatever. So, from just a tiny, tiny amount of dried feces, we can, in a matter of hours, basically sequence any gene we want, carry out paternity analyses, individualization analyses, all sorts of genetic analyses. So now, in fact, we can collect so much data. The problem has become how to analyze it. So in fact, analyzing the data has become more important in some aspects than the wet lab work. This ancient picture from 1989, how many of you were born after? After 89. Yeah. Okay. So in 89, I was a grad student sitting at the bench at Harvard University, manually sequencing DNA. For every A, C, G, and T, I had to carry out a separate test tube reaction, run them out with radioactive dyes, expose them to X-ray film, develop the X-ray film in a series of tanks like you see people developing photos in the movie, and collect a very little amount of data. In fact, I collected 700 bases of a gene from the mitochondrion called cytochrome oxidase subunit 2 in 15 different species, and was given a Harvard PhD for that. It took me 3 years to collect 700 times 15 different species bases of data. I now have undergraduates who collect 200 times that in the course of a single semester. The next project that I am putting in these techniques have moved so rapidly and advanced so far. The project we hope to complete, we just got funded and we hope to complete over the next several months, is going to generate 50 gigabases of data by December. 50 billion bases of DNA sequence from baboons is what we're going to be working on. And that's just completely unheard of even 4 years ago. So the advances of going from 4 separate test tubes to a single tube, to running it through slab gels and then capillary electrophoresis and looking at these, we are now in the phase where this amount of data is almost nothing. We are now using machines that generate billions of bases basically in a single